Pilot, whenever you're ready, we can get going.
Okay, so that ship move is going to be at 050, is that right?
Yeah, 050.
Let's dial it in.
So for your awareness, we are going to be moving at 050, and we are starting the transect now as they just sort of finalize their lighting configuration. So for Mike, Amanda, and Mike, we will have about 5-minute clips of all of the data that we'll be collecting during during this long transect. So just be prepared for that. They'll be clipped into 5-minute batches, so you can handle it a little easier.
I think it's dependent upon the ship's GPS.
Yeah.
Great, thanks.
So
Okay.
I would say move along it.
So, Mike, I'll turn it over to you just to sort of however you normally proceed.
Talk about speeding up, but for now, I think this is good.
Well, what's our goal here? Are
Okay,
we
thanks.
covering distance or just time? Because I don't think there's no need to go at point 2.
No, we won't. No, we want to be down at point 1.
Yeah, because we're just looking for stuff. So we don't.
We want to be moving point 1 through the water, ideally.
Yeah.
And the watch lead will be telling you to look at a specific organism if once we see one.
So Roland, or do we have Tara? We got Tara.
Yeah,
do you want to go ahead and zoom in like partial, like
I
halfway?
got the ship going, point 1 over ground. That's right now.
As long as it seems like we're actually moving the same direction through the water.
Yeah.
Yeah.
Okay. I think this is point 1 over ground for the ship.
Yeah, unless you have
So I
a
think this
lot
is
of
pretty
targets.
good.
My suggestion is that the pilot
we're
just
at
automatically
450
stop
meters.
and try and image anything large that they come
So you guys
across.
will let us know if we need to change depth.
Yeah. Okay. Okay. We'll pass that on to them.
Okay.
So, as watch lead, why don't you tell them that they should image anything that looks significant.
All right, Pilot, this is watch lead.
Go ahead, watch lead.
So did you guys hear the request from shore?
I did not.
Okay, so they have requested that essentially anything that you come across that looks like a target, let's go ahead and image it.
Okay.
We're not anticipating a bunch of big targets, but if you see one, let's grab it.
Okay, so no potential whales in this area, huh?
No, but if we find another one, I would love to get it on ROV video.
All right. Well, Tara, you got the better monitor over there, so if you see something,
Roger.
let me know. Don't be shy. Let's just zoom in on this white thing here.
Sure.
We have 1 hour left.
Nope.
All right, you should have your first target in front of you in just a second.
This is gonna be a video workout here, I think.
I'm not sure if that one's
All right. Yeah, let's go back up partial.
Ooh, that's something that's moving. Oh.
We're at. There's a little bit of a current coming starboard to port. I mean, you can see the particles in the water going in that direction.
We can turn into
Everything's going
turn
by
into the
very
current.
quickly right now.
Pilot.
What's that?
We can
Everything's going
adjust
by
the ship
very
move
quickly
to
right now.
get you into the current to a certain extent.
Yeah, I mean, so are we? What is the itinerary here? We're just at this depth, and we can.
There's something.
Doing decimal.
Yeah, just trying to get a
moving through.
transect
Yeah.
and see as much as possible and get as much detail of anything we see.
I'm noticing quite a bit of starboard to port current, and it's going to make it kind of difficult if things are just whipping by side to side.
Why don't we
Can we
try
turn
to
into
actually
the current?
turn into the current?
Yeah.
We'd have to turn the ship into it, but
Yes.
yeah.
So.
Works for us.
Let's try to get a current here, and then I'll adjust the ship move.
Okay. Let me see if I can.
If it doesn't go into
So
shallow
I'm looking
water.
at the EK60 data. And it looks like is that, and if we can just confirm it, it seems like there might be a layer
Back to-
at about 450.
Look at like a,
I think I'm looking at that correctly. That seems to be
Maybe
something
one.
real. Actually, possibly on both.
We are getting returns from the vehicles, right?
Yeah, so
Yeah.
I think you're actually seeing the two vehicles, one above the other.
Yeah, the
I see those
two little lines.
two constant lines there, and then okay, so I see a layer above at about
I think it's
350.
somewhere around maybe
okay.
110 or so. Let me
So those straight lines, Derek.
It seems like there's a
Maybe a
good
little
layer up where we started. We could come up pretty quick. I'm serious.
Yeah, it is looking like there's a layer just shy of 400, maybe 380.
You're at 80.
That looks pretty good.
Yeah, the
090.
So we can see how this goes, and
Yeah.
if we're not
Why
seeing
don't
much.
we
can move up.
adjust to 090?
Well, I mean, right now we're adjusting the ship's heading and so on, the ship's direction. It might be a good time to change depth if you want
Yeah.
to. It's not very far.
090.
It's like 5 minutes to get up there.
What do you guys think on the phone?
bridge over now.
That sounds good to me. What do you think,
Can we
guys?
adjust that heading to
I'm fine with
bearing
that.
to 090?
So what's the depth? 380? 3.
So Don, co-pilot.
Yeah, go ahead.
We're going to
Yeah, it
adjust
looks
our
like-
being to head into
Or
the current.
almost 400.
So we have an easier time managing.
just about 400.
We're changing the ship's motion to
Yeah, that's all right.
090.
I'll talk to my pilot.
What do you
Thanks. Yeah, you can do that.
340.
Keep going.
Yeah, that sounds more like it.
Oh yeah, because I
That's
see the line. 340. And what do we have it here?
Good copy, bridge.
Pilot, do you read me?
About the same.
Go ahead.
Yeah, we want to while you're adjusting
340.
things, we want to also adjust our depth and come up
Okay.
to about 340 meters.
Okay.
Can you do that and stay in this configuration?
Yeah,
Good.
I sure can.
Good.
Yeah.
Yeah. So focus
So
on
let's
the
wait
line.
for
It's looking like 340 meters.
Is that
the,
should move in?
Yeah,
depth
so
we're
he'll
shooting for.
change bearing. What he's going to do is I think he's going to hit present position.
Okay.
So you might swing just a tad. We're only
Okay.
going decimal 1.
Yeah.
And then he'll throw it in. So, yeah, just keep an eye on the vehicles, but everything should be fine.
All right. Well, Don, let's start coming up. They want to go to 340.
340. Hauling in.
Very close to where we started.
Yep.
Yeah.
All right. It's good
Thank you,
practice.
pilots. We appreciate it.
Thank you,
Going
Bridge.
up is way easier than going down.
Yeah.
Okay. So this is part of the reason that we wanted to run the EK60s, though, during these midwater transects and take advantage of our new sonars, is that we have the ability to adjust on the fly. And when we see something
Okay.
like a
We can
new
come up
layer
at 30
or a new
meters
layer,
and
we are
maybe
able
like
to adjust to
20,
that and
25.
25.
So we'll
move
slow.
accordingly. Hopefully find some more exciting things.
Yeah, there we go.
So, Mike, we also have a CTD sensor on the vehicle. Would you mind, either Mike or Amanda or Mike, talking a little bit about sort of how you would use the CTD data for this kind of, for characterizing the water column?
Okay, this is Mike Ford. We take a look at the CPD data for a couple of different things. I think we also use the CPD package on the wire that can be cast immediately before the dive as well, and that would help identify any details in the oceanographic profile. So any interesting details in temperature, salinity, or oxygen that we might see vertically in the water column that might give us a tip to relate that to some of the biology, so we keep an eye on that. And over time, it's interesting as we take a look at these observations of biology in the midwater, it's very good to have these kinds of parameters that we see these organisms at certain temperatures, potentially at certain salinities, and certainly at certain oxygen
maybe
levels.
bring the
So
starboard
that's kind of what we're using it for today. For every observation that we make in this very long midwater transect, we'll be able to connect those observations with the streaming data coming from the ROV and just add the environmental component to our biological observations to make a much more robust record of the exploration.
All right.
yeah.
Thank you, Mike.
So we're halfway there.
Add that the two things that we're seeing on the CPD that are really important, other than the depth, is temperature is physiologically really important to the animals, but also dissolved oxygen, because there are places in the world where waters at these depths are depleted in dissolved oxygen. And what we're seeing from the data from the ROV here is that there's plenty of oxygen in the water here. That becomes important when we start making comparative observations in other places where there isn't as much oxygen in the water. It also becomes important because global climate change is expected to change the oxygen distribution substantially, as well as the temperature distribution in the water column.
We're coming up on our target depth.
Large expansion
So I'm going to have you come up to 330.
330, roger that.
A problem for the
How's that look on the light bar?
Thank you, both mics.
Swing on.
You can slow down just a little bit.
It's the CTD package and the dissolved oxygen content is something that we regularly collect, but I actually don't know that we've had anyone really explain that while on the line, so it's really great. So thank you so much.
Yeah, looks good. Thanks.
Okay, so we can go ahead.
So we are currently approaching 350 meters.
See that yeah, so.
And we are headed right into that layer. So, and I just saw a siphonophore blow by.
We're almost in position. Watch lead.
Thank you very much, pilot.
I have to say, watching the trace of the two vehicles and on the same screen as watching the scattering layer is really neat and extremely useful. If we get tons of targets here, this could prove to be an excellent device for us to use over and over.
Okay. All right. So let's both stop now.
All right, pilots are
okay. All stop.
Stopping and getting ready to start our transect.
All right, pushing out.
So, Mike, we have talked a lot.
Tilt this Sirius camera up a bit
recently about sort of the unknown nature of the water column, and you provided a bit of content about that just now. But one of the things that we've spent a lot of time thinking about, and we've been working with Amanda Netburn to sort of flesh out a little bit more, is what should water column exploration look like? So one of the things that we're doing here today is testing a new method, testing some technology, and seeing if this works, if this is the right way to proceed. So would you talk a little bit more about along those lines, please, while we wait for the pilots to get ready?
We are currently in the transect.
All right, perfect.
We are moving. We are at 340.
Derek, is it possible that the vehicle return is sort of at this point masking the biomass return?
If you can maintain
that's sort of what it looks like. Now that the vehicles are up at the same depth, and we sort of saw the same thing when we were originally at a similar depth.
It's either that or we're scaring them away.
Oh, there's a siphonophore.
There's some things that could be scared away by us. Hopefully.
Oh, sure, of course, but
yeah. But yeah, I think a strong echo like that would just maybe mask.
Pilot, would we be able to zoom on that siphonophore?
Of that hydrophone, do you know?
I'm not sure off the top of my mind.
All right, thank you.
Some calculations on that.
Yeah.
But I'm gonna.
Well, that siphonophore that was just at the top of the view, that is an example of the capability of an ROV being able to see the siphonophores in a nice long configuration. They typically would get broken up or disturbed by the net. So it's nice to, I think that's always been one of the things that we've observed on these midwater dives, is seeing these long and delicate animals just as they are in the water column.
Yeah, you can zoom at will, Tara.
Yep, sounds good.
You see something.
Not sure what that is. There's a little tiny fish.
There's a little fish going in on him.
Might not be a fish.
He's definitely squirming.
All right, we have another target coming up.
Oh, he just swam away.
Okay.
That
There's
was
something
shrimp.
in the picture. Going for it. We could almost use a bit more light right in front where the cameras are. When I zoom in, it gets pretty dark, and I'm full light.
So, Mike, was that what we just our first target back there? Was that a Keating-Nath?
Hey,
I think that was. I was looking quickly for dive codes.
Upper swing arms in anymore?
Yes,
Are
it was
they-
a Keating now.
Thank
Yeah,
you, Mike.
They're all the way in, like inward.
And it should also be noted that we greatly appreciate the
they're
three of you guys
perpendicular.
staying
They're
up
all
very
the way
late. I know
out,
you all
aren't they?
are on the East Coast,
The
and this
upper
is
ones.
very late for you. So
Come back
thank you so
further.
much for joining
Can
us
you check
today.
to see if we can bring them in a bit? Yeah, bring them in a bit. Yeah.
We're all really excited to be doing this.
Yeah, this is a pretty cool way to spend the night.
Tara's saying that she needs more light.
It's like stargazing, but underwater.
Upper swing arm stow. There we go. All right.
Yeah, so I guess they are all the way down. So yeah, just bring them in a bit. Maybe that'll help.
Okay.
How's that a little more, Tara?
That's on the port side.
That's
the other
one
over?
side, yeah.
It's a little better, yeah.
A little better. All right, we'll
Yeah, that is better.
go to the
Cool.
starboard side.
Thank you.
You want to zoom in on that, whatever it is.
And that would be also stow coming in a little bit.
Oh, too close.
Any thoughts on what that is?
Okay,
What did you say, Amanda? Sorry.
I was just wondering if there are any thoughts on what that is that we were seeing in the center of the screen.
That looks like, it's marine snow. I mean, there's a lot of material in the water. It could be from aggregations from, larvations and so forth, and
So Don, just keep an eye on
stuff kind of falling or sitting in a
I'll
layer.
be drifting with things as they come by.
It
And if I
looks like a
start
lot of
drifting
this material
backward
is
too much,
so
why don't you
small.
pull up on the winch and
Roger that.
come up a bit? Yeah, I'll try to keep out well ahead though, so that we don't have that
Okay.
issue. But
Thanks, Mike. Yeah, it looks like what we were looking at was,
Yes,
was
it's a
a fair
lot
amount bigger than the rest
okay,
of
cool.
it.
Let's go
but,
partial turn.
Probably just something that had aggregated.
Oh, wait a minute.
Yeah, you're right. It was a pretty good clump of stuff. I was always looking for aggregations like that. Sometimes can be like a larvation house or something. It has a good image. But that looked like it could have been something there.
There's something there, bigger.
I see what you see.
We're gonna go in partial.
Mhm.
We have another target coming up in
Siphonophore.
just a
Maybe.
second.
Look like a
Oh, you can kind of see. Looks like it's cilia.
Ctenophore.
Behind me,
Is that a big ctenophore?
Roger that.
Ctenophore.
backing up?
Oh, that's so cool.
Or a septum, or I think it's velellum.
I'm hauling in.
And it's that structure in the center that it helps you determine which group it's from, depending on the shape of it. So that's the key part to image.
Oh, that's so cool.
That's beautiful.
This is assisted, like the Venus girdle, isn't it?
Yeah, that's great.
Wow, that's incredible. So something like that would have definitely been destroyed by a net.
Yeah.
Oh yeah.
Weird radial effect there, huh?
But Mike, is that very compressed, or is it just the angle that we're looking at that makes it look that way?
Maybe we got the
Oh,
lights in
the
too
shape
close.
itself?
Yeah.
Yeah, they're long and
Like a
kind of
string.
ribbon-like like that.
Camera's
long.
all the way down. Yeah.
They're really cool.
Back.
Yeah.
Okay. Great partial.
That was a great video, pilot. Thank you. And video, nice job.
Compress a lateral plane, so they're really long and stretched out, and that sort of tube structure we're looking at in the center with the mouth,
Thanks, Don.
Roger that.
stomach, so on.
Uh-huh.
Pushing ahead now.
I am pushing
Yeah.
ahead.
Very cool.
There you go.
Thanks.
Something that's
Give you a little more slack.
I don't know too much about the sort of the ecological advantage of having such kind of a long surface area, unless the use is to set up some sort of a feeding current along that entire body. That's very, very interesting.
Yeah, I wonder if they're using the flat body itself as a mechanism for capturing some particulates. You know, they don't have the long retractable tentacles, so I think it's the body itself, kind of like flypaper.
Yeah, kind of, sort of like the maximum development of, you know, you see things in like Bolenopsis with big lobes, or even if you look at Bathychoroe and you sort of just develop that body form and just much bigger, and the whole thing becomes this big flapping mass. It's really, really cool.
Ooh.
Salp chain.
Yeah, it might be that we have the light bars pointing inward. Tara said she needed more light directly in front of the camera. We can play with that a little bit.
You want to try pulling the back a little bit?
Sure, yeah.
All right.
So,
angle it more from the side.
looks like we have a salp here, or is this a siphonophore?
Oh.
So, is that supposed to be?
How can you tell the difference between a salp and a siphonophore?
Can you pull up on the winch, co-pilot?
That's a salp chain there.
So what's the difference?
Roger that. Coming up.
Sorry, I know you're juggling stuff.
Siphonophore would have a lot of dangling tentacles coming off of it, and it has swimming bells concentrated at one end.
Keep an eye on it.
This is a colony, and each of those little dots that you see on it is an individual animal in the chain.
Oh, cool. That's good to know. Thanks.
So, yeah, so to follow up on
Okay, I'm going to
that,
adjust the upper swing arm.
There's differentiation
port
of the
side
parts.
right
So the swimming bells and the feeding bells, or the feeding individuals that look very different and actually serve different roles. Whereas the salps are
move that
really
one a
kind of
little bit,
identical
and
organisms
now we'll try
that
the
are
other
all
side.
on the
See what
chain.
happens.
Great. Thank you, guys.
Just one more contrast. A siphonophore is an idarian. It's a carnivore, so it's using its tentacles to capture something and ingest, whereas the salps are tunicates, and they're basically filtering particles.
Yeah, it's just another
Looks
one
like
of
more
those.
detritus, but up to the upper left, that might be something else.
Thanks, guys. Looks like we have another distant target,
He's floating down.
but it may or may not be able to stay in focus.
Yeah, he just floated down.
He's escaping.
Yep.
There'll be no escaping here.
Does
So
it look
guys,
like those,
on
light
shore,
rings have disappeared
we
now
have
that-
Looks better, yeah.
the pilots still have the light bars out a little bit, but how are you, compared to some of the other midwater transects that we've done where we have not had the light bars out, how are you finding the image quality now that we've had a couple targets?
I think it's been pretty good. We've had some really clear bodied animals, and I think it's been okay. The Venus girdle is always going to be tough to have really super contrast. I think the South looks pretty good. I don't know. What do you think, guys?
Yeah, that South looks great to me. The Ctenophore was really hard to see, but I think that might just be how that organism is. But if we saw something else like that, I don't know how quickly we can make the adjustments, but it might be good to play around with it when we actually have an organism like that in view.
On that?
Hold the light bar. Where's it?
We can do anything. I'm just not quite
Oh.
sure.
What is that? Coming up on it.
Got a new target for you guys.
Oh,
Oh.
he
This
just
is
jumped away.
a very large copepod.
Whoa, monster copepod.
Gorgeous.
I mean, we can adjust both the angle of the light bars, but also we could start turning off the lights that are on the main light bar across the top.
I'm not going to really see it very well, but aha.
Oh, we've got another one of those Tina 4s.
Yeah, it looks like we have our chance. This looks great, actually.
That's great imagery of that center structure there. That's great, and that's critical. Oh, yeah, that's great.
This is much easier to see, I would say, than that last one we saw. That's incredible.
This is closer, I think.
Wow, look at the way those... Is that cilia at the edge of them?
Yeah.
Yeah, that's cilia all along the edge. The Tina 4s we're used to looking at, you see the 8 bands that go from the, quotes, top to bottom, the oral to the aboral, whereas here they're sort of aligned along the long edge.
Wow, that is incredible imagery, guys.
Am I looking
nope,
down?
I'm full iris.
You're looking good.
Okay.
Yep.
Can you crush the blacks and turn up the iris anymore?
The iris is full.
Okay.
And if I crush the blacks...
That is a weird effect.
Yeah.
Okay.
But they're already...
All right.
They crushed.
Let's let it go.
Yep.
I'll push it
Time to push ahead.
Yep. For the central part of the stomach.
That is...
So pilot and video, have you seen that effect before?
I have not.
I don't remember it either.
Can we... I don't know, should we try stowing the light arms, or is science happy with having them out?
So on shore, guys, we're noticing, and I'm not sure if you're seeing it too, sort of concentric circles around when we try to go in and focus on things. So that is not something that we've noticed before when we've had this light bar stowed for these sort of beauty shots while in the water column. Would you like us to try and stow them to see if we can get a clear picture? So instead of having just the sort of center of the frame in focus with those sort of frames of rings of distortion, we'd have the full...
It's not distorting it's not a focus issue.
Oh, it's just light?
It's light reflection.
Oh, okay.
Inside the dome. I think...
Maybe Jim had a good idea.
Yeah, and I think it makes the imagery less beautiful, but I don't think it's necessarily affecting the quality of our ability to see what we're trying to see.
Okay. So what do you guys think on shore?
I didn't see that effect on my computer, but I'm certainly open to trying it without, seeing how the quality is.
All right, so we have one vote for try it without, and what about Mike and Mike?
It only takes a couple minutes to stow.
And Scott, you're on the line too.
Yeah, let's try it without.
Okay.
Yeah, you should have confidence in your video engineer to make that choice, as they've been focused on video and lighting for many, many years.
So Tara, I think that means you get your wish.
Well, yeah, I agree.
But, I think science probably has as much experience. We're usually not
I think the upper light bars are what I would stow, not the lower ones.
I think a group decision would be...
The upper light bars are farther out forward.
Let's try it. See how it works.
All right, Don, we're giving you practice with the hydraulics today.
Roger that. What do you want to do?
We're going to stow
the upper lights. Stow the upper lights.
Stow the upper surround. So I'm going to zoom in. Let's zoom in, Tara. And Jeff is going to try turning off the uppers. See if we still see that effect. Just a bit.
Yeah.
Did you turned off the lower ones as well?
Would be better for anything that was
Might even try turning off the ones on the light bar and turning back on the ones on the swing arms.
Turning off the mains?
Yeah.
Yeah, try turning off the mains. Oh. That just does a different...
That's not bad, really.
Oh, you want to know what that is?
Let's zoom in, Tara. Zoom in
Just,
tight.
just SAL.
Yeah, we're still.
No.
This is strange.
For some.
Oh, really?
When I go full zoom, I lose the light. Just
Do you see it on your monitor, Tara?
Oh no!
Oh.
Oh, maybe the monitor.
Casey, I think it's our monitor.
It's the monitor.
Yeah.
Oh,
What
well.
are they seeing? What
Apparently,
are they seeing,
the distortion is.
Gee, I must have really been
The light rings are only are not visible on a good monitor.
Let's get the lights back on then.
All right, never mind, guys.
Explain why we're not seeing.
Sorry about that. I mean, it's
that clears that up.
Quite a big light ring on our monitors here.
Good.
So, well, I'm glad.
Oh, cool.
Oh, cool.
Yeah.
There he is.
Gorgeous.
Is he not? Nope. He's hanging out down below. There
Will
he
not
is.
be anticipated.
Well, thank you everyone for your patience and for experimenting with us.
Turn off the lights while we have an organism to see.
No problem.
Which looks better.
Hey, do you want to kill?
Yeah, so Casey, I'm just watching on the public feed on
Which one's?
Maybe the upper swing arms.
Been frozen since you went ahead and turned the lights down.
Like that.
I don't know if it's my iPad I'm watching on or what, but just letting you know.
Okay. So we haven't. It may have been just a little dark. We're not really moving all that fast. But if you restart your browser, that should help.
Yeah, right. Let's bring it back up. I think we can.
Video wants them back on.
Okay, I'll give it a try. Thanks.
Okay, thanks. And if you're in the event log, you can access the high resolution, low latency science feed.
All right.
Yeah, not yet.
Next
I'm outside walking my cat. You can believe that. She demanded it.
Gotcha. Well, we appreciate you joining us.
For the people at home, we've seen a couple of heating NAS, and people have been referring to them as that, but they're also known as arrow worms. And they're a separate phylum, a major group of, an evolutionary group of animals, all to themselves, basically. And they are voracious predators, and they're a really important part of the deep water pelagic ecosystem. So seeing them is an important thing that we're coming across.
Thanks, Mike.
Let's get that, wherever that guy is. Zoom in. Zoom in tighter. Oh, no.
That's about full.
How is that full? It's pretty far away then.
Shrimp.
Is that shrimp?
Looks like it, yes.
That's what I saw.
That's my vote too. Hard to get a good view on it.
Gonna hold partial.
That's fine.
Just look for another target.
Ooh, is that a fish? Zoom out. Zoom in. That's not him. Let's zoom out.
So for your awareness, guys, we are about 30 minutes from needing to wrap this up. So we're about halfway through our midwater period.
Oh, there's something.
Oh, okay.
Thanks, Casey.
What about that jelly-like? Oh.
Backing up, but I think he's gone.
So for those of you at home that are just joining us today, this is NOAA ship Okeanos Explorer, and we are exploring the water column today. And we just have another target that should be coming into view for you guys soon.
He's going upward. Zoom in, quick.
Yep, pilot, we're getting about directly overhead.
Okay.
Push out.
Yep, pushing out. Thanks for keeping an eye on that. Roger.
So any idea on what that last target was, guys? It was a quick one.
But yeah, it looked to me like it was a kind of short south chain.
Okay.
Yeah, I agree. That looked like south. It was tough to figure out if it had anything that would give it off as a siphonophore. We'd have sort of a contrasting brightnesses on each end.
Trying to correct for the motion.
Yeah, I think you're right. I'd say it was probably south.
Or if it's the vehicle moving. So zoom at will, but just let me know what you're doing.
So as I had started before, this is NOAA ship Okeanos Explorer, and we are currently exploring Midwater just off of Mog. We are within the islands unit of the Marianas Trench National Marine Monument. And this is the only portion of the National Marine Monument that has protected waters as well as protected submerged lands. So we're spending some time while we're in the area making sure that we get a good characterization of some of the animals that live and inhabit those protected waters. So we have another target up here, guys.
Wow, that's another great chested penis. Look at that. That's great.
Backing up.
Like, do you have a sense if these are typically very common at these depths or common in this part of the ocean?
Well, we've seen a bunch of them, so I think they are fairly common. But they might they might be something that we mostly see with ROVs and with cameras. Much easier than probably what has been sampled in the past. Oh, that's great! Look at that. Do you have a guess on the approximate size of that?
Just a bit wider.
Yeah.
Because that might give us a tip as to what species it might be.
All right, let's try this again.
Could we,
The lasers aren't going to be much use.
we're not going to, you don't think we'll be able to get lasers on that?
I don't think we'll even be able to tell when the lasers are on them. Well, maybe, but we could try. I mean, there's no downside to trying.
All right, he's
It's out of light, yeah.
Pilot, this is Watch Lead.
Yep.
If we see another one of those and you have the opportunity, would you mind trying to line up a laser shot? I don't know that it's going to work, but we can try.
There's the lasers. But, I mean, unless the dots sweep over it precisely. I'm not even sure
it's going to be a lot of refraction
on that creature.
It'll show up a little bit, but...
Yeah, okay.
So folks on shore, you guys should be able to see our lasers now. They're 10 centimeters apart. And is this useful to have them on as we sort of move through the water column?
Oh, yeah.
Coming back around, Don. Roger that.
I think they're useful if we get lucky and we have something fairly big that we get the clear hit with the lasers on the body of the organism. It's tough to do for midwater, but I can't think of any other better option.
Okay. So we'll leave them on. Thank you, guys.
I don't think they detract from anything, so I don't see that being a problem.
Yeah, if the camera was instrumented, we knew the focal length and the focus distance, we could back it out, but that's a lot of ifs.
All right, Tara, keep your eyes peeled.
I'm on it.
So I have to say, our pilots are doing a fantastic job holding this depth and moving and focusing and trying to find all of the
There's something. Yeah.
all of the targets that we're coming across, and we are exceptionally lucky to have video broadcast engineers who can help get this kind of incredible imagery.
Got pushed by the prop wash.
You guys are making it look easy.
So if folks look back at the EK60 sonar view here, I think it seems like we have some avoidance behavior perhaps going on.
Yeah.
The layer we moved up to is
moved down
below us.
Wow.
So it's possible that our vehicles are just such a strong return. We're masking biology nearby, but I think there's also a lot that is avoiding the vehicles due to the lights and noise.
There's a cell or something.
If anyone on the phone has observed that before, I'd be interested in knowing.
I'm sorry, could you...
On the EK60 and run while we were doing midwater trawls, and we've been able to actually watch the layer in out of the way of the net before it got to it.
All right, we have a nice, beautiful new target for you guys to look at.
Ooh, there we go.
It's got a big head.
Wow.
Hold that.
That's great. Siphonophore.
Oh, that's cool.
Priya, right? Yep, that's great. Look at that. 2 swimming zones at the top.
This one is a Cali-Cochrane siphonophore.
Is it that, or is it Priya?
I think Priya is a type of Cali-Cochrane.
Oh, okay. I'm sorry.
So there are two types. Two major types of siphonophores. There are the Fisonex siphonophores that have a kind of air bladder at the top of them, and this is called a nematophore. Helps them kind of keep their buoyancy in the water column. And then the acoustics in the EK60 backscatter, they reflect similarly as a fish that would have a gas bladder. And this is one of the reasons why it's really important for us to do these kinds of surveys, because there can be huge layers of Fisonex siphonophores that from the surface we interpret as fish because they reflect so similarly. The Cali-Cochrane, like the one we're seeing, does not have that kind of bladder in it, air-filled bladder, so this would not reflect very highly in the EK50.
Is that Tara? Trying to move closer to it,
yeah.
but...
I'm going to come out just a touch.
Okay.
It's getting just a bit too...
That is a beautiful shot. That was so cool.
Incredible.
All right.
Okay.
Yeah. I'm going to push out ahead.
Roger.
Nice job, Tara.
You too.
Did I hear that the layer we were chasing is at a different depth now?
That is correct, pilot.
What are they reading now?
Deeper.
Back where we were originally.
And just took a random sample of the biosphere. What they would catch is one of these jellies.
The word is out on the
midwater.
I have seen the layer scatter as the ROV approached before.
That actually is an amazing image. The EK60.
So, Mike, I actually missed that. I apologize. I was talking to the pilot. Would you mind repeating, please?
What I was saying was that because most of the planet's biosphere is deep midwater, and most of the animals are gelatinous, if somebody from somewhere else came to this planet and wanted to find out what kind of life was here and just took a random sample of the biosphere, what they would catch is one of these jellies.
That's fascinating.
Yeah,
I never really thought about that way.
Okay, coming out a little bit.
So what was that we just zoomed in on, guys?
Not sure. That might have been just a random piece of something.
Here, here.
Yeah, I think we would have had to try to follow it to the other end.
Interesting. Turn the lights off for an hour. Be interesting to turn the lights off for an hour and see what it does.
Yeah.
Ooh.
Should we just stay in?
We're just going to cancel the dive tomorrow and hang out here in the dark.
Wait until we see the sonar layer come back up to us, and then we'll turn it back on, and then we'll do it again.
See if they run away.
How quickly can the sonar tell us that they're avoiding us? Sonar response time.
Yeah, he's gone. Oh, no.
They're such small little creatures, they sure swim quickly.
It's a very good observation, Tara.
It's something on the lower screen, but it just went off.
The problem is I'm pushing ahead to get ahead of Sirius, and then I slow down real
So
quick,
looking
and
at
the
the
backwards prop wash
EK60, I
pushes
mean,
them away.
is it that the layer moved, or we just can't see it anymore? I mean, I'm looking at about 350 is just the vehicle that I see, and the next return seems to be below 400.
Right. We've been talking about the fact that the scattering layer seems to be running away from us consistently.
So Derek had mentioned before, too, that it's entirely possible that we are masking the layer. So I wonder if there was a layer at what we were at now when we first started at 350, and then but we masked it, and then we went down, and we could see the layer again at 350.
It's going to be interesting to watch what happens as we come up from there.
Yeah.
Because I don't think we're masking it.
Yeah, that would be a good test.
I don't think we're masking it. I think it's avoidance behavior. Something is going away from where we're making noise and light.
I think so too.
So the other experiment we'd love to do but don't have time for is turn off the lights and see what happens.
Are we making much noise?
Not necessarily the lights that are doing it.
Right. No, I realize, but we can.
A lot of noise.
Right. But it's easy to test the lights. It's hard to test the noise.
There's something down on the bottom right.
Well, I'm thinking, maybe we could try turning off one or two of the Sirius lights.
I don't think it's going to make enough difference to worry about unless we turned them all off and did that for an hour, which just obviously isn't practical.
Okay. This is very steady. Do you want to zoom in, Tara?
How are we doing on current pilots?
On current? It's pretty negligible here, actually.
Great.
I'm off stick right now. I'm not moving too quickly.
Okay.
And things are pretty stationary in the frame. Zoom in, Tara, and see if we see something in the distance.
Great, thank you.
That's full zoom.
We're so steady.
Copepod? Look at that. That's great imagery.
Zoom close. Or, sorry, focus close.
Oh, right here.
Yeah.
Ooh.
Beautiful.
Oh, wow.
Good spotting. That's nice.
Any idea what that is?
It's moving quickly.
Yeah.
I have no idea.
Oh.
just like anything I've seen.
Yeah, that's crazy. I'll never look at that.
All right, let's zoom out a bit.
Ah, there's one. All right, zooming in.
There's a couple down.
Let's zoom out.
Pilots, we got about 15 minutes left.
Okay. Copy.
So shoreside, we have about 15 more minutes.
Great. Thanks, Casey.
So no thoughts on what our little spiky friend was?
For now,
if you want speculation, it might have been a baby siphonophore.
Oh.
They start out looking different.
I didn't know they changed so much when they got older. That's interesting.
That's just a wild guess.
It's all right.
Okay, there's something sort of top of the screen.
Sure.
It's pretty little though.
We're going to do a quick pilot change.
Roger.
Thank you, pilot. So some of the things that we've been doing here on the Okeanos Explorers, we have a largely new team. So we've been working with different members of our engineering team to get them trained up. So who is our new pilot?
Levi.
Levi. Is this Levi's first time piloting? Flying? No? Okay. Sorry. Sorry, Levi.
Oh, you're muted.
Ooh.
Pilot for about 20 minutes, so this is a good practice for me.
Well, welcome and good luck.
Yeah.
Oh.
Hey,
what is it's moving?
This is a larvation.
I was going to say larvation.
Inside its house.
So
larvations create a music.
Okay.
Larvations create a mucus house that collects. They use it to filter before they get their food.
So I've seen the houses before, but I haven't seen one with
that's
it
the
inside.
larvation fluttering inside.
Oh, cool.
Yeah. They're helping to kind of keep pumping that water through the house. And this guy was pretty ready to abandon his house. Pretty dirty, I would say. And they abandon them quite regularly and then very rapidly build a new house.
It's the ultimate green building.
And when they do that, the old house sinks down into the deep and transports carbon and food source down to the deep. It's an important method that.
Yeah. So you're in auto depth. So.
Yeah, I'm not just.
Yeah, it's a good concentrator of organic material, and that's what a.
But yeah, just so you know, if you do try to adjust depth, you're gonna have it run away.
Directly related to the
so
south chain.
Oh, okay.
If you want to go up or down, just
Thanks,
bump
guys.
it up, bump it down, just for half a second. Okay.
So
Change the set
how
point.
do they
But
know when to change their house? Can they detect that particles are filling up on it, or do they just do it on a certain interval?
I don't know.
What makes a larvation change houses?
Was the question, when do they beat the tail?
No, when do they change their houses? Do they just do it on a regular interval, or can they actually detect when it's time to change the house?
Well, I would imagine the house has filters in it, and then once they get clogged and the particulates can no longer reach the mouth, then it's time to get rid of that house and build a new one.
Nice.
Oh.
I've actually seen them abandon ship when the house got.
So one thing to
around
remember
by
too
turbulence.
is when things are moving up and down on the screen, that's also due to your forward-backward motion. So as you move forward on something, right, they're going to appear to move downward, and as you move backward, they're going to appear to move upward. So you can adjust.
They look a
you can
little
adjust
more
with your camera and then
apart
fine-tune it
than
with
some
your
of the other ones we
forward-backward
saw
motion.
earlier.
Okay.
Try and get tight.
Maybe 3 of them?
I don't know. They all seem to be moving together, though.
A little longer though down there.
Just trying to look at the pulsing.
Zoom out just a touch.
Yeah, maybe. Oh. I don't know why I thought there were 3.
Almost smart target. I probably pushed forward.
No worries. We'll forgive you this time.
Go for this little 4 dots here.
That one?
Yeah. Try to get into view here.
SIP.
Yeah, pilot, we're coming over top of you, just about over top. Okay. Need to push ahead.
Copy that. Video you could pull out real quick?
Sure.
That's good, thanks.
So, Amanda, would you call it Freya because it's a much more rounded
Nice job.
morphology than rocket shape?
I don't know the different siphonophores very well beyond the Calicopteran and Siphonac, so I'm not sure about that.
Yeah, no, I wasn't 100% sure.
Yeah, sorry, I need to.
I would always have thought that the Calicopteran were all really kind of rocket shaped.
Ooh.
Nope. Yep.
What's that, guys?
Oh, great. Big cell.
It's a very big salp.
Yeah, so this is the solitary life stage of the salp. It's just one single individual.
You got me at 82 degrees, looks like.
And it's
so I'll have to keep moving here in about...
the number of bands, if you can see the bands around the tubular body, that helps figure out
10 seconds and I'll boogie
what kind of salp it is.
When you're good, video.
I can see it just tumbling. That's great.
Cool, thank you. All right.
Nice imagery, Tara. That was gorgeous.
Nice job.
Yeah, that was cool. Thanks. Wow.
That's great. That's really great. And like on the siphonophores, it's possible that I was wrong on that, so I'll have to look into it some more. I don't want to be giving out misinformation.
Amanda, can you type the name that you're using into the event log?
Yes.
Thanks.
Amanda, I think you're right. I think Freya is a Calicopteran.
Philpot, can you bring your Zeus up to like 45 degrees?
Thank you.
Coming up. Roger that.
It's always good to have validation.
And wrong ID. That's just unheard of for Okeanos operation.
Okay.
Sorry, can you repeat that?
Is that where you like to fly?
Yeah, and then, yeah, you're going to bring it down.
Oh, yeah, you're probably not wrong because that Pryor would fall under that. So you're cool.
Don't listen to Amanda. They're just giving you a hard time.
And Amanda, if you're wrong about it, join the ranks of those of us who uttered out lots of bad ideas.
We are just collecting the initial round of data and the initial set of observations. So even just a guess is helpful to someone who can eventually prove us all wrong. Whoa, that's cool looking. That's a cool copepod. Are you guys able to see that?
It's a polychaete, not a copepod.
It's a polychaete with those long...
one of the pelagic polychaetes, the Tomopteridae. I'll write it in the event log.
Oh, cool. I didn't know those existed. Are they predatory?
Yeah, it finally came on my screen. I think the Tomopterid.
So can you tell us a little bit about those? I've never heard that phrase or that type of organism before.
It's a pelagic polychaete. Spends all its life up in the water column.
So for the general audience, polychaete is a kind of worm, and most of the diversity of these worms is benthic. They crawl around the bottom, build tubes, and so on. And we see a fair number of them when we're on the bottom looking at the communities. But there's one family that's adapted for living in the water column, and there are a few other genera that are restricted to the water column.
Nice.
Cool. Thank you, guys. You learn something every day. That was fantastic. So sometimes when we've done these midwater transects, we see things that are transparent, and then we see some with some really bright reds and sometimes some blues in them. Today we've seen mainly clear and translucent organisms. Any thoughts as to why?
Sorry, Casey, can you repeat the question? It was about why we're seeing so much translucent.
Yeah, so sometimes when we've done midwater transects in the past, we see some red organisms and we see some that have a little bit of blue in them. And today it's been pretty much exclusively transparent organisms. So I was curious if one of our water column experts would mind talking for folks at home about why would an organism that lived in the water column be transparent?
Yeah, so basically transparency is camouflage for organisms in the water column. So just like we're having trouble seeing, or not trouble, but it can be hard for us to see these animals. And the same thing for their predators. So they're able to hide by doing that. And it's transparency and gelatinousness, which is basically just a really high concentration of water in the body tissue. It's something that's evolved in many different parts of the tree of life and lots of different families of organisms, phyla of organisms. So we see, the jellyfish, but also many of those pterographs are gelatinous, and the tunicates, which are you're seeing the salp. And these are really organisms that are not closely related, but have all evolved
10 minutes
transparency
50 meters.
Because of that
2 or 3 minutes
ability to hide in the water column.
Thanks,
so
Amanda.
Call in the 15-minute call.
That's great.
3 minutes.
No problem.
And then you'll have a couple minutes.
And I don't know if we're not seeing some of those other things because they swam away in that layer. Or if it had something to do perhaps with the optical properties in this particular patch of ocean.
My guess is we're not deep enough.
So
it's daytime where you are, and most of those red organisms are strong vertical migrators.
Okay.
And they would usually be a lot deeper in the water column during basically midday, down where the light is just this very dim blue, and the black, the red absorbs the blue light, doesn't reflect any of it. So that's their way of camouflaging. But because they're strong vertical migrators, they wouldn't typically be this shallow during the day, I don't think.
Yeah, that's a great point. And much of the work that I've done has been in the California current, which is much more productive water, and therefore much darker at these very same depths. And so I think we do collect a lot more organisms with that kind of coloring, with the reds, darker colors. But the water here is much clearer than it would be along the west coast. So it's going to be a different fauna. That's interesting. It's great to see.
So hopefully over the next couple weeks, we will be doing a couple deeper midwater transect dives. So when we do those, maybe we'll see some of them then.
It'll be very interesting to see.
All right, guys, we have about 1 minute left of our transect. So if hopefully we maybe get one more target. If not, if you guys want to sort of start wrapping up our discussion about some of the things that we saw and maybe some highlights.
Coasting now.
So,
Okay, pilot, we're going to do a swap here. I'm scheduled to be on deck.
Copy that.
Roger.
all right, I'm going to put in the 15-minute call out.
So I thought the ctenophores were really great. That was cool. We saw a few of them, and they were.
All right, watch lead.
And I think the imagery we got was great.
I'm going to put in the 15-minute call out, and we're going to start setting up to come up to the surface. Bridge, Nav. Can we please get the 15-minute
Sorry, Mike, please continue.
call out for recovery?
I had to chat with the pilot real quick.
Just maintain, and we're going to get set up and I'll through the water.
Tested cenophores.
Are you happy with your current heading?
Very interesting. We got some excellent video of the three targets that we did see. It was really interesting. Actually, I think your question is really interesting about sort of the balance of organisms, and I was kind of thinking about that too. That's really.
We'll keep it in a position move. We're going to get the vehicles set up, and then I'll have you come ahead.
Kind of
Thank you.
One long transect at what looks like almost precisely the same level the entire
time. So let's start getting set. I'm going to have you set up.
That's been interesting.
So if we look at your turns,
to take a look at compared to
Jeff will
the multi-level.
Take care of that. So Sirius is set up. If you look at your turns, which direction do you need to turn D2 in order to straighten it out?
Anything else?
Just to look at the EK60 traces
that's the number of turns between Sirius and D2.
Off of the Midwater guide
one is the turns
to see if we can detect other layers.
So you have a positive turn, so you need to turn to port.
Yeah, that'll be very interesting. We can leave that up on feed 3,
so
at least for a little bit, as we start the move. We are going to do the dive planning call. Probably we'll give it maybe 5 minutes or so. Derek just stepped out to go prep, so we will give it a few more minutes. So if you guys are interested in staying on the call, we will be back in about 5 minutes, or rather I will be joined by some people in 5 minutes. And so we will be talking about our next dive site, which is Hadel Ridge, and the dive after that, which is Ahi.
And you're going to turn around.
So
get your heading
as we are starting, we haven't quite started to ascend yet. So, but it will be very interesting to see what happens with that layer as we move up into the water column.
And then once you're at the bottom, you're going to keep pushing ahead slightly.
I also want to say it's an excellent opportunity to explore the Midwater. I really appreciate the time to do it on these dives. I think this is a very unexplored part of the ocean, and every time we get to spend time here, I think we learn more and we generate some interesting questions. So yeah, thanks. This is really great.
Yep. And then.
All right.
Yeah, same here. I appreciate you spending time in Midwater.
Push ahead. Come up.
Well, I'm glad you guys were able to join us. Thank you so much. And again, thank you for joining us so late at night on the East Coast.